First of all, my name's Jacob Kalina. I'm a UGA Extension agent in Lowndes County. Thank you all for joining us here at the Southeast Regional Fruit and Vegetable Conference. Without any further ado, I would like to introduce our first speaker, Dr. Oliveira, Vegetable Crop Precision Agriculture, and I'll let him have it. I don't know if this, yeah, it's working.
I'm going to take it off. Yeah, just stretch my legs. Good. Good afternoon, everybody. Thanks for coming. Thanks for attending this session. Promise you're not going to fall asleep. And if you fall, I'm going to drop this clicker on you. I'm kidding. As Jacob said, my name is Luan Oliveira. I'm the assistant professor and precision ag extension specialist with the Department of Horticulture at UGA. And I'm here because I have a couple projects with vegetables. Actually, it's a lot. My main objective here is to bring information for you guys about the technology and robotics that we have been working with. And as an extension specialist with 75% extension appointment, my job is actually bring that information to the grower. So if you are a grower and you want to know more about what we're doing, shoot me an email. I'm going to have my information as well at the end, and I'm going to be glad to answer any questions you have. Without further ado, so that's the Vegetable Crop Precision Ag presentation. Here's my team. As I told you, we try to touch a lot of ground, and we're working on AI, we're working on mechanization, crop quality, autonomous spraying, and several other pieces of precision ag inside of vegetables. So the first one I'm going to present here is the spraying drones. So it has been a buzz in the past 2 years. A lot of people are adopting or increasing the use of spraying drones in agriculture. And for vegetables, it will not be a lot different, because I think that the place for spraying drones is actually in vegetable fields. So this is a carrot we were spraying. The trick here is how we can get the best coverage for those crops. Each one of them are taller, spacing is different. The leaves are different as well. So how can we make the best application? First thing you have to do is to measure it on the ground. How much is the effective swath? How much ground that drone can cover? So in this example here, and I'm sorry, I'm going to be trying to bounce around. Like, some people are looking that way, some people are looking this way. In these two graphs, you see that we tried 5 GPA and 10 GPA. That means gallons per acre. And here with no addition of surfactant, which is adjuvant, and with adjuvant. And we wanted to know how far is my application swath. They call it swath. My effective swath, which means that if I'm looking to have at least 40% coverage, I'm talking about in between 6 feet and 16 feet of application. So I have 10 feet of effective swath, right? With this drone, we're at 10 foot, applying, I would say 10 gallons per acre only, because at 5 gallons, my effective swath decreases a lot. And that's in the ground. So we just placed water-sensitive paper grounds, water-sensitive paper in the ground, and then we found out that the effective swath at 10 gallons per acre is 10 feet. But how does that work on plants? We have a lot of products, and the reason we are using 5 gallons per acre is that the most majority of the fungicides, which are the products that we can apply with a drone, I'm not going to put herbicides there because not all the vegetables are I would say hybrids to tolerate those herbicides. So this is a list of registered fungicides for aerial application, because here's a trick here. If the label says that you cannot apply through an airplane, you cannot also apply through a drone, okay? And on that label it says you can apply it aerially. And the minimum that you can apply is 5 gallons per acre, 2 gallons per acre, or some of them 10 gallons per acre. And then we tested this in cucumbers, yellow squash, bell pepper, tomato, carrots, and pretty much two other crops, two other vegetable crops, and these are the results. These graphs are pretty busy, but the main take-home message here is that 10 gallons per acre is better. You can cover with 10 gallons per acre right there. Following the row, you can get like really good coverage in the top of the plant, up to 60%. On the we did 6 rows of tomatoes. So this is row number 1. This graph is wrong, I'm sorry. 1, 2, 3, 4, 5, and 6 rows. And the 3 middle rows, they were good. We got good coverage there. 60% coverage, droplet density, which means the number of droplets inside of a square centimeter. But at 5 GPA, we did not have that really good coverage as we found with 10 GPA. What is the issue here? Efficiency. A drone only carries about 15 gallons. That's my drone. It's one of the biggest on the market, which means that I'm going to have to send that drone in. The drone is going to spray about an acre, come back, I'm going to have to refill it, swap batteries, and send it again. I don't see an issue on that, but I always compare ourselves with row crop growers, and the row crop labels, they allow you to go up to 1 gallon per acre, and our labels don't. So that's the difference right here. Okay, that was the drone spraying part, and I'm going to talk a little bit about robotics. If you see this really busy picture right there, this is called the 2024 Crop Robotics Landscape. All the companies included in the segment of robotics for agriculture are here. Not all of them, but pretty much all of them. This number increased from near 200 in 2022 to 400 in 2024. There's a lot. And what scares me the most is not even that, is that the number of robots being sold or included in catalogs is increasing a lot as well. So just look at that red square right there. On harvesting robots, and I got scared when I saw that number. The number of harvesting robots sold in between 2022 and 2023 increased by 1,200%. And that was because a company out of California, they developed this machine to harvest strawberries, and they sold a lot there. But in general, the number of robots for agriculture increased from 1,400 to 2,400 in between 22 and 23, and that means a 73% increase. How we're going to take decisions if we have a lot of those on the market is like you go to Walmart and you see that whole aisle of bread. It's impossible to pick one, right? What is the best one for strawberries? Which one is going to fit my operations the best? So thinking about that, my group and I, my team, we started doing some work on robotics applied for specialty crops. And the first one is that little robot right there. This is called XAG R150. It's an air blast sprayer, really small one, 50-gallon tank. First thing we measured, we wanted to know how far that spray pattern would go, which crops could we cover with that. And this is the app, this is the controller actually for that robot. And in the app, you can select the direction of those air blasts. And it swaps, flaps horizontally or vertically. So we want to see in a fixed position how much is the coverage. And then we did 25 GPA, 75 gallons per acre, and 100 gallons per acre at different degrees. You see, we were at 130 and 160 right there at the horizontal. Company said that we could get about 40 feet coverage there. And indeed we got, but like the most, coverage, like the high concentration was in the first, I would say, 20 feet right there. 20 feet for each side, which is not bad. But when we used the robot in the way it's marketed, it didn't work really well. You see, it moves horizontally right there. And we did for different crop spacing. This is a simulation, right? 6 feet tomato, and then 4 different heights, 8 feet vineyards, 4 different heights, and 12 feet for blueberries with 4 different heights. And And here are the results. Actually, the only good coverage we got was in 6 feet for tomatoes, using that flap on. So me and the guys at the lab, we said, what is happening? We did on the static there, it was supposed to go at 20 feet. Because of that, it's too slow. That air blast is too slow. So it's kind of going up the plant, coming back. So the coverage is too variable inside of that field. Then we decided to do a fix-it on those 3 heights. And then we got good coverage at 6, I would say. Some good coverage at 8, but not at 12. We got some here at 12, like 15% coverage as well. And let's see here. This is a fixed bar again with, and that was let me see that one. That was 25 and 50 GPA at 130 degrees, and this is 160 degrees. Coverage didn't change much, but you see here, we got 75% coverage. Oh, that's the bar attachment. That's not the air blast. So the bar attachment is that robot is like a Lego. You can put an air blast, you can put a bar, a boom sprayer, horizontally or vertically, and then you can adjust the output on a hydraulic pump. That's not on the controller. On that one, you can do on the controller, but this one here is a hydraulic pump. It's really hard to actually calibrate that thing. But that bar at 6 feet gave us 70% coverage. And more or less at 8 feet for vineyards, it would give us more or less like 25%, 30% coverage as well. Okay, that same robot, you can attach a mower on it. We did some trials. We run some trials on that. It has 2 batteries. When you purchase that kit, it comes with 6, I believe so. And each battery takes more or less 10 minutes to charge. And the efficiency of the robot mowing was actually more or less like 45 minutes to 1 hour. So it gives you enough time to charge the batteries. The robot is going to come back. You swap the batteries and send it back to mow your fields. Limitations, it doesn't work inside of orchards. In open fields like that, it did work, but inside of orchards, it did not. Because those are called GPS denied environments, so it did not work inside of the pecan or blueberry orchard. Another technology we've been exploring here in the lab is called Solix. This is a solar-powered robot. This company is from Brazil, but they're out of Midwest, and they developed this robot to run the whole day on solar and do spot spraying on the crops. And they are doing a lot of row crop work, and this was actually the first application on vegetables ever of that piece of equipment. And what we're doing right now, I don't have any data on spraying. We're just doing training for them. We're training their artificial intelligence model to know what a broccoli is, to know where a lettuce is against the weed. This is an application report from that robot. You can see here, like on the first trial we did, the model was not accurate enough. We were able to save like 59% of the output of the chemicals doing that spot spraying, and it generates a map of the weeds in the field, like how much times it opened and closed those the nozzles. And then coming back, leaving the robot, drone precision ag side. Another thing we've been working on is mechanization. So I was a county agent in Nebraska for 2 years, so everything I know was corn and soybeans before coming to Georgia. So first thing I wanted to work on Vidalions because I really liked them back when I was living in Nebraska. They're really sweet, right? So I said, I want to work on that crop. We went to the field to measure how much time those field workers take to transplant by hand, because everything is done by hand today. And then what we found was this. We found a lot of plants laying on the ground. And after that, we wanted to know the impact of that of those plants laying on the ground, and I'm going to show you some results. I'm pretty sure I have it here. But in percentage, this is plants laying on the ground. Yeah, the gray bars here. Per acre, in the morning and afternoon and late afternoon, they leave about like 10 to 13%. Actually, the average is right there. Yeah, it's about like 10 to 8 to 10% of those onion seedlings just laying on the ground. And then if you're an onion grower, you know, there's a belief that those onions are going to make it. Even though they were just left laying on the ground right there, they're going to make it anyways. What we found is different. We found that there's a huge difference. We set a trial in 2023 where we placed 100 seedlings on the ground, and 25% of those were partially transplanted, or just laying on the ground, as I showed in the picture. And the control, the difference in between the properly transplanted seedlings against those ones was about like 10,000 pounds per acre difference. So right now what we're doing, we are trying to implement mechanization into onion transplanting. We're not reinventing the wheel here. We're just getting commercial transplanters that are used for different crops and trying to adapt them to make them work for onions. So we just placed the first trial on the ground back in December. We're going to collect the results and see how that goes. What we know right now is that field workers are faster than transplanters. However, the quality of a mechanical transplanting is better than a field worker, because I'm placing the same all the seedlings at the same depth and at the same spacing. Of course, the field workers are doing the same, but they're not being left laying on the ground and everything. So this is the data. So we tried 0.3 miles an hour. That was too fast. We could not keep up with the seedlings. We tried 0.20 miles an hour. 20 miles an hour. It was doable, but it requires more training. But at 0.15 miles an hour, we were pretty comfortable on placing those seedlings inside of the mechanism on the transplanter. And if you think about how much and how much time it takes for me to transplant 1 acre at 0.15 miles an hour, it takes about 9 hours. It's too slow. It is slow. I agree with you guys. But if we compare, we had 2 studies. We had 2023 and 2024. If we compare to the slowest person in the field, because we measured like 70 people this year. If we compare with the slowest person, it's the same. With the average person, we're losing about like 2 to 3 hours. With the fastest person, we're losing a lot. But we're gaining quality. Another part of my research is AI, as I mentioned. We are not like, this is not something we developed. This is a product on the market already. It's a platform. I flew a drone right there. Actually, really low, I flew it, okay, so I could get more resolution. And then I'm going to zoom in that image, and we're going to be able to see those bulbs. Onion market. Is there an onion grower here? Okay. So an onion market, it varies by size. You know, I think that it's more important for them to have jumbo onions, which USDA says that there is a specific range of sizing for those. And we wanted to know if these new platforms, these are AI-driven, if we could count and if we could measure those onions only using drone images. And last year we did this preliminary data collection. I don't have the results yet. I'm going to have more, I would say, concrete and accurate results this year. But as you can see, the AI was trained to identify the onions, and some of them are being either skipped or under-measured. And I didn't do nothing. I'm going to show you guys in the video right there. How much time I still have, Jacob?
About 30 seconds.
Oh, yeah, sorry about that. you can come to me later. The only thing I want to show is a model that we developed in-house to predict onion yield. So we flew the drone 6 times every other week, 60 days, 90 days before harvesting, 75, up to 15 days. And we could predict the yield of onions with 71% accuracy just by flying the drone. And of course, training the AI models on that. With that, I think I'm on time. Appreciate your attention. If you have any questions, I don't think I have time for questions, but I'll be here. Thank you.
Dr. Quezada? Okay, yeah. While I get his set up, if anyone has any questions, I got to switch over the laptop. So
those tomatoes, they were, I would say, between 3 and 5 feet tall. Yeah. 60. Yeah, no, I got 60% coverage on the top, so that decreases a lot when you go to the bottom. Yeah. I would take 96% coverage in the bottom. No. At 10 gallons per acre, that was like the best coverage we got. Like at the middle plant right there and the top, like right below the drone, that was 60% coverage. Alrighty, thank you. Oh, okay. Appreciate it.
with that, I'll hand it over to Dr. Quezada. Am I saying that right?
Sure, yes, that works. Hey, everybody. Thank you for being here today. I know it's Saturday afternoon. We just had lunch, which was great, by the way. But, you know, it's the weekend, so I'll try not to make it too boring. Hopefully, it's going to be a good presentation. Is that the pointer right there? Yeah. Cool. There you're good. All right, so what you guys are going to see right now is some of the efforts that one of my master's students did at North Carolina State University, which was basically playing around with bed designs and how to improve the fumigation efficiency of the methods that we have right now. So a little bit about North Carolina. I guess it's better if I stay somewhere in here. North Carolina, we do produce tomatoes. Most of our tomatoes are going to be to the west of the state, between Henderson County, Rowan County, and Bucombe. I'm probably butchering that thing right there. But those are where our tomatoes farmers are going to be, most of them. We do produce about a 1% or 2% of the whole production of the United States. And we do use plastic culture system, which is the most common system that you're going to find in the South, right? So there are many steps to producing tomatoes. And my lab, we focus a lot on solanaceous crops and brassicas. So we do touch on most of the things, the boxes that you see there. Just for today, we're going to talk about bed architectures and establishment. So here's how a commercial, traditional tomato system looks like in North Carolina, especially early in the year. So we're talking 24 inches wide on the bed and 8 inches high for the traditional bed. That if you're looking at an acre basis, most of the rows that you're going to have are going to be at least 300 feet long. So if you calculate the volume of soil you're going to be using for that setup right there, 8 inches by 24, that's going to be about 400 cubic feet of soil. The shape is trapezoidal. Farmers tend to use black or white traditionally, and we tend to work that in 5 feet center to centers. How does that look in the field? Just like this. There's a bunch of numbers in the pictures. The idea here is that I want you guys to see how the distance goes from the center of each bed to the other, and what size of aisle you're going to have based on the dimensions of the traditional beds. In this case, we're going to be having about 3 feet of aisle where your work is going to harvest. The wheel of the tractor is going to go in some cases, at least for some sprayers where you don't have that aisle every 6 rows. But anyways, once you do the numbers for a full acre, that's going to give you about 8,712 feet of actually bed in an acre. So that's just the basics of the traditional beds. Our planting densities is between 2 feet in-row spacing, single row, or 1.5 feet. Some farmers do want to go a bit closer. So your planting density is going to be between 4,000 and 5,800 plants in an acre. Now, in Florida, this is all coming from Florida, they decided to start working with changing the architecture of the beds. And this work was originally started with Dr. Chukla at the University of Florida. And since then, it has been moving up north in the United States, especially in the east side. What we're talking here is tall and narrow beds, which basically means you will change that architecture from 24 inches as a basic to 18 and increase the height of the bed. So we're going from 8 all the way to 12. And the premise is that because you're narrowing the bed and increasing the height, the distribution of the water bulb that is inside, because you got your drip tape right in the middle, is going to be better, close to the shoulders of the actual bed. If you have a sandy soil and you have a bed that is 24 inches wide, then that water will have to laterally move all the way to the shoulder, and sometimes that doesn't really happen. So in terms of the water distribution, the results that he had found in Florida is that in these taller and narrower beds, the water moves better, which is good. The shape stays the same, something that in our case we tend to do, and also some of the big farmers in Florida is shifting from from black to white and reflective. This is some of the data that we collected in Virginia Tech when I was there. We try out different mulches in tomatoes, and this is the effect of having in summer-grown tomatoes reflective mulch compared to black. So the color does affect your final yield. And just so you got a more graphical, description of this, this is a thermal images of the trial that we had in Virginia Tech. You can see here the multiple mulches. This tree right here, those are the blacks. Then you got your white and your reflective. So, and the black kind of looks like an oven when it's on, basically. And the reflective and the white, this is going to be the white, and the reflective kind of looks black. And it's because all the light is being reflected, and the camera basically thinks that it's looking at water. And that's the way water looks like on the thermal camera. So our farmers, especially in the east, are going definitely for white and reflective. We're skipping the black mulches unless you got a really cold year and you're planting extremely early. You really don't want to do black. And this is just another representation of that, but then with the temperatures of each plastic mulch, being the black right here on the top, at the peak of the day, we were getting temperatures of 120 degrees Fahrenheit in the soil under the black plastic, compared to the white and black, which and reflective that you can see right here near 100 degrees. But those 20 degrees right there are the ones creating that difference in yield that you saw in the other ones, which we're talking almost 8 tons in difference in yield. But anyways, back to the beds. We are definitely working with white. We're still maintaining the 5 feet center to center in our beds, which again is going to give us the same feet per bed of bed per acre. And then you got your planting density. So all that we're changing here is the actual distribution architecture of the actual beds. So taking this technology and going towards the fumigation side, given that in Florida most of the work on yield and water distribution have been done, we thought, okay, how can we take this and try to integrate this to the ways that we fumigate the soil? Because at the end of the day, most of the fumigant is going to be injected as a gas, as a liquid. It's going to become a gas, and it's going to stay under the soil. So the actual shape that you have for your bed might be influencing the way that gas moves in the system, right? So those are the questions that we had. How does narrow, taller beds design impact soil fumigation efficiency in tomatoes? Are chunk and drip fumigation equally effective, especially in this new architecture? And does adding soil surfactant to the fumigant mixture enhance the overall effectiveness? This one at the end was mostly related to a question that I had some years back, which was, if I add a surfactant to the mix of the fumigant, can I reach that shoulder of the planting bed that often doesn't get fumigated? Have you guys ever fumigated tomatoes before? Anybody here? Or any crop for that matters. All right. Have you noticed how on the shoulder of the bed, if you have a lot of nutsedge, that thing is going to come again, but it's not going to be in the center of the bed, rather it's going to be in the edge? That just means that the water with the fumigant didn't really get there, right? So if we try to break that surface tension of the water and try to improve the movement of the water, ideally the fumigation would be improved too. So these are a list of multiple fumigants. There are there in rating for the effect for nematodes, disease, and weeds. We went with Picklor 60, which has been pretty well, reported for nematodes, good to excellent for diseases, and also fair to good for weeds. And also the reason why we went with this one is because there is a EC formulation that is allowed to be applied through the drip. right? So in this case, we were applying basically the same component in 2 different ways, chunk and through the drip. Here are our treatments. We took the traditional bed configuration, which in our case, we just maintained the width. We actually increased a little bit of the height. So we call that the standard, which was 24 by 12, without fumigation, with chunk fumigation, with drip, and with drip plus surfactant. And then we took the compact, again, with the same fumigation treatments, and at the end, with the surfactant. And that's what we got to the actual field. These are just a few pictures of how we, the things that we did at the field. We got a special bedder that is adjustable that allows us to have the different bed architectures. Pretty expensive, but if you're a farmer, you don't need to do that. If you get the right size that works for you, it's going to be cheaper. Fertilization, pretty standard for our conditions in North Carolina, and we were irrigating about 0.17 inches a day of water. It was sandy soils. It was in the middle of the summer. It was pretty hot. Anyways, here's what we got. In terms of disease, in terms of weeds, we didn't measure weeds before the fumigation because there were no beds, right? So there's no something before the fumigation. But after the fumigation, what we noticed is that all the controls for chunk, drip, and drip plus fumigant did pretty well compared to the control, which was shooting weeds through the plastic and through the hole to the planting hole. So in terms of weed control, the treatments did fairly well, all 3 together. Both at 6 and 8 weeks, we were seeing the weeds at the controls and still pretty clean for the rest of the treatments. In terms of the initial nematodes populations, we went ahead and before we put the beds in the field, we took, it was, I want to say a field probably double or triple the size of this room, and we took something like 120 samples. So it was fairly representative of what was going on in that field. And these are the averages that we have for each of these particular plots. So we identify where we took the samples and where these plots landed, and then we assign that to each particular plot. So we had a basis of where the nematodes were. And this is what we got after the fumigation. The controls, of course, stay fairly high. And then for the chunk and drip plus fumigant, the control was, again, fairly good. This is across all the populations of nematodes. We had to combine them because the numbers for some of them were not high enough. So we just all put it all together, and this is how it looks like. So the control for nematodes between chunk and drip plus fumigant was fairly good compared to the actual control without any fumigant. If we look exclusively at the fumigation, without touching the bed architecture, it was pretty clear that chunk and drip were doing fairly well, especially for pre-cloxicity at the rates that we were evaluating, which was 175 pounds per acre, compared to the control. Now, looking at the yield, because if you had the pests and you got the diseases and everything, you might control them, but if it doesn't affect the yield, then what are you doing? What are you doing, right? So we look at plant height, and in general, we didn't see differences in plant height. This is just the general trend of our plants growing throughout the season. Moving towards the actual biomass, we didn't find any differences across the biomass of the different treatments. We used the cultivar Jolene, which is a fairly tall plant, high biomass, if you ask me. The fruits are not too large, but it was a good number of fruits. And in overall, the controls and the rest of the fumigants, the biomass for the plants were doing fairly well. Now, what I want to pay attention here is, rather than the actual fumigation, is the type of beds. Because remember, this one is the compacted, this is the standard, this is what we tend to use, this is the new thing that we're trying, and overall, they were all doing the same. This is important for what I'm trying to drive at the end of the presentation. In terms of the actual yield, again, between the standard and the compacted bed, there was no significant differences for the actual yield that we were getting. This is not we saw the same trend basically for fruit per acre. And we got a pretty high number of fruits. The actual final yield wasn't too high because the fruits, in my opinion, were somewhat smaller. We were getting an average of about 120 grams per fruit, which is basically a fairly small tomato. It's not a huge tomato. But again, there was no difference between the standard bed and the compacted beds. For the fumigation part, in terms of the the actual yield, comparing fumigations across the two beds, what we found was that the controls, of course, were putting a lower yield compared to the fumigating trials, and we didn't find differences between the chunk, the drip, or the drip plus the surfactant, meaning that the drip fumigation or the chunk fumigation will work fairly well if you're trying to use it, especially in sandy soils, and at least in North Carolina for tomatoes. A few extra data points that we collected here. So the last thing that we did measure was water distribution, because we were trying to explain how the fumigant was moving in each of the beds. And something that we found was that, depending on the height of the bed, especially in your sandy soils, and for this particular setup that we had, which we were using 0.27 gallons per meter, 12 inches for each meter at 8 psi, we noticed that if we irrigate more than 30 minutes for a bed that is 8 inches high, we will go deeper than the actual profile, the cross-section of the bed, which is not ideal, because anything below that, you're basically leaching nutrients. Yeah, the roots are going to get there, but the majority of the beds are going to be in that cross-section right there of your bed. In our case, for the tall beds, because we were using 12, that would allow us, for this particular setting, an extra 10 minutes of irrigation per event without really reaching the end of the bed quite just yet. So it's just an extra little thing that you could do with taller beds. This is what we mean about improved distribution of the water. This is just, and I'm about to finish, this is just a 2D animation of how the water distributes in each of the beds. This mark right here is the 8-inch mark, and of course, each of the examples are 12 inches high beds. We put different multiple soil water sensors, and we try to create this in a profile. What I want you guys to notice here is how the accumulation of water in the bottom here, if you over-irrigate, compared to how well distributed in the compact bed that water is. So something at the beginning is that you might notice is some extra water here in the shoulder, and that's because the majority of the roots are going to take this direction, almost like a triangle, if you will, given that the plant is going to be on top here. Here, the roots were reaching the shoulders faster, therefore the consumption of water was better distributed across the beds. And to finish, this is a quick comparison. Another thing that you can do with the compact beds is that because they're compact, we can move from 5 feet to 4.5 feet, because at the end of the day, if you're trying to maintain that aisle of 3 feet in the middle, because the width of each of the beds is just smaller, therefore once you combine all this, you don't need as much space. These actually translate to potentially an 11% increase in the number of beds that you can add in an acre. The only issue is that the 5 feet is pretty much defined by the size of your tractor. So to compensate for that, what we need is a 3-row or 6-row bedder that can have actually that distance between the planting rows, so you can have that 11% increment. This is something that we're trying to secure this year, so we can do the testing. I don't foresee any reason why we shouldn't see the increase in yield, because the first few trials, there was no competition for light that we noticed for. So these are the preliminary conclusions. We're going to be running this again this year, and also we're doing the same trial with different bed architectures for strawberries, so hopefully I can bring that information for you next year. This is just how they look in the video, the beds that we were testing. Last thing, we're also trying to create maps with the distribution of the different parameters of the soil, because if you can get maps for your soil and you see differences in the textures of the soil especially, you can start to create fumigation zones, if you will. A sandy soil is going to behave different than a clayey soil when it comes to the distribution of water. Therefore, if you can identify that beforehand, then maybe we can identify particular rates that apply better for one soil rather than the other. With that, I appreciate the time. My name, again, is Emmanuel Torres, and if you want to find me, that's the way to do it. We put a bunch of extension publications with this information, and I can answer maybe one question, I think. Excellent. Thank you. Thank you so much.
We'll go ahead and move on to our next speaker, Dr. Nankar, and he's going to talk about pepper breeding, some cool stuff going on.
Thank you, Jacob, and thank you all for staying till the end. What I'm going to do is basically just walk you through some of the things we did last year. as this is a brand new vegetable breeding program we started recently. So before I jump into paper breeding, in addition to the paper breeding, we are also taking into consideration some of the other crops which are important for Georgia and Southeast US, including snap bean, tomato, and partly onion. So our priorities has been mainly on what growers really need. And since it was a brand new program, I thought I need to use the opportunity to get to know from growers where we should exactly focus. And we did the concept mapping survey last year. So we asked like 7 set of questions, including demographic information, then what are the preferred 3 varieties they grow, why they like those 3 varieties, and what are the desirable and undesirable characters associated with those 3 varieties, where we should focus on the breeding components, and then what are the major diseases and faced, and quality traits, and how to take it from there. So in this survey, we got quite a interesting information on those commercial varieties grown by growers, what are those traits that we should focus on. And this has been a very useful information for me to undertake into upstream breeding process. So beside peppers, we also did it in tomato, and then snap bean, and then broccoli. So this survey has been a key component in setting up our breeding pipeline. And I appreciate all those who participated in this survey. And if you are still interested, there is a poster outside and there is QR code. So we are utilizing the opportunity to go outside the Georgia also and take some of the inputs from Southeast growers from different states if you're interested. So taking it from there, coming back to the paper story. So in paper, basically, these are the 4 major important issues, including Phytophthora, bacterial spot, anthracnose, and root knot nematode. This consistently came in during the interaction with the growers. So we're going to focus on Phytophthora as a primary target, and then anthracnose, bacterial spot. Beside those 4 traits, some of the other diseases, which we will consider later on once we have major findings on these. So insect resistance, paper weevil. Paper weevil is kind of very interesting, and I have had a discussion with the colleague from Europe. He said, if you are going to breed for paper weevil, we are not going to like it, insects are not going to like it. So there is a trade-off, and we have to work on the strategy, how this really work, and we can find potential resistance. Then thrips, green peach aphids, then white flies a little bit. So beside insects, fruit quality is super important. And when it comes to the fruit quality, the fruit composition, since it is a nutritional constituents which are important, so we have to take some of these traits into consideration, and we'll focus on it. But more than fruit composition, the fruit morphometry, like the size, shape, color, these are super important that we have to be mindful in the breeding program, that we have to take them into consideration, because nobody would like to have a bell pepper which is not really suitable for the targeted market segment. And then standard horticultural traits, yield, doesn't matter how great variety we have, yield has to be there, because we have to maintain, we get the capital out, and we maintain the profitability. Then earliness, again, it's a specific market niche. Mechanical harvesting, it's a big one. And I really don't know what direction we'll be going in that direction going forward. Plant architecture, it's associated with the mechanical harvesting. And then foliage, especially when it comes to the paper, especially bell pepper, the scarredness. So we have to be mindful of foliage, and we have to take them into consideration. So this is a big story, and based on this, the big picture, what we did, since it's a new program, we got the seeds from gene banks domestically in the US and from outside. So first we had to do the seed increase. And we use 1 plant per accession in the greenhouse, collect some of the information on it, and we were able to get some of the good seed increase so that we can do all these experiments what I mentioned, and we needed those seeds. So TED has been very resourceful in making sure that we were successful. While we were doing seed increase, we made sure that we are collecting some of the key information that will help us to characterize the germplasm. And that could be our reference point on what exactly we are looking for while we are doing the breeding. When it came to stem anthocyanin, because that's a standard in the commercial industry, and that we have to be ensure that we are taking into consideration the fruit position, whether the fruits are going to be in erect or pendulum or a drooping position, because that is associated with the harvesting component of it. Then stem height, blossom end rot. That's another important component that sometimes the growers are feeling they are having issue with. Then plant architecture, that's a standard horticultural traits. The fruit diversity. So we have 500 accessions, and 500 accessions represent 169 countries. And we have tremendous amount of diversity from extra large bale to standard bale to mini bale, even a tiny bale. And then we have banana peppers, then snack type, cayenne, serrano. We call pumpkin shape, but Ted calls it pancake because that's a standard in here. So we have all these kind of diversity in our collection. So what we did while we got this all information for simplifying these 500 accessions into more categories. So we divided them into mini sweet, banana pepper, mini bale, bale. Even the picture might look for mini bale and bale same, but it's a position of the camera where we took the picture. Then Italian friar, and then specialty peppers. While we did the seed increase, we also decided that why don't we use this opportunity to collect some of the information on a large scale and grow them in a breeding field trial. That's where Ted's team was very helpful us to set up the trial in the field. And we have had 2 replications. It was not really like a complete trial as such, since we were late almost 6 weeks, but still we were able to make it through the season, get the seeds. Some of the traits which we scored based on the commercial breeding industry would do in terms of the fruit set, then flowering time, maturity, whether it's lodging, the heat tolerance, and then the plant architecture. Even though we didn't get all the data for all traits which we were envisioning, but we got some of the data on germination, then plant vigor, days to flowering. This days to flowering is very important because that allows us to select some of those early ones that we could include them into breeding. And having the early maturing could offer the growers a potential to grow during for the premium pricing. So that's one of the thing, and we included. And the days to flowering, what we are seeing here is from seeding to flowering, not from transplanting to flowering. So on an average across all these varietal groups, there were around 81 days from seeding to flowering. Then there was a plant height. Again, it was taken before the flowering stage. Stem height, then lodging. Lodging is associated with the mechanical harvesting. So we are taking this trait into consideration while we are looking at long-term picture, downline, 5 years or 10 years. Because in Tifton, in summer, sometimes there are windy days. And when there is a strong fruit setting done on the plant, if there is a strong windy day, the paper plant can lodge, and that really helps us to get this data. And sort of like would allow us to select some of these promising candidates from there. So beside the conventional breeding aspect, we are incorporating molecular breeding here. When I say molecular breeding, it means there are some molecular markers that we can use to screen the whole collection and know from these 500 accessions, okay, out of 500, there are 20 accessions. They have the phytophthora resistance. So we don't have to necessarily wait for until screening the whole thing. We are going to do the screening, but we want to use these tools in order to save the time and expedite the breeding process. So we have been setting up the lab, molecular lab, for doing the marker-assisted selection, and we have started doing marker-assisted selection for Phytophthora. So there are total 32 molecular markers available. Basically, this is like a pinpointing one marker for each trait in each gene location, and that helps us to screen the whole collection. So we are running like 16,000 reactions on a PCR. It's like 8 hours running PCR for almost like 10 days and getting that information. But we are very excited about the findings from the marker-assisted selection. So that work is currently going on, and once we finish the screening for Phytophthora, we're going to do the same thing for anthracnose, bacterial spot, root knot nematode, and that would bring us to the major part of the breeding is the pre-breeding. So when we say the pre-breeding, it means from this collection of 500 accessions, we know that there is one accession that has a resistance against Phytophthora. So we are going to take that one resistant accession, and then we have another accession that does not have resistance, but the growers love it here. And that accessions or that kind of variety gives the growers potential high price, high yield. And currently, we are making the crosses for Phytophthora, and we are planning to do in bell pepper with a 4-lobe, standard 200 gram size. That's the ideal vision that we would have potential bell pepper. So we are doing that for Phytophthora, and then beside the Phytophthora, we are also making potential breeding crosses on bacterial spot. And we are hoping that from these breeding aspects, we'll be having potential segregating populations that we will be screening next year and see if we have potential breeding candidates that we could release in future. And we are looking maybe around promising candidate in 5 to 6 years. Or if I get some of the infrastructure, I could expedite that process to maybe 4 years. So breeding takes time. So whenever I talk to growers, they say, hey, when are we going to have a PCAP resistant bell pepper, Georgia bred? Like maybe 4 years. They say, really? Yeah. So it takes time. So that's the vision we are carrying forward. And I would like to go in more details, but I hope I'm 7 minutes. So especially I'd like to talk on this pre-breeding aspect is, right now we have around 20 candidates which has a resistance against Phytophthora. But that material is basically from different parts of the world. That's not necessarily suitable for Georgia. So right now we are trying to figure out a strategy. If we make those breeding crosses, would they be adapted to Georgia? And that with that strategy, with the prior data, we are trying to identify if we can have some of these candidates for Phytophthora. Second story that is coming with bacterial spot. We have around 7 candidates that has a bacterial spot resistance, and these are the bell peppers. They are mainly for European segment, and then some of them are for the South Asian segment. So we'd like to see how we can take it and breed those more suitable for Georgia and Southeast Asia. So beside that, I'd like to take this opportunity to thank all the funding agencies. We have been receiving a lot of support from Georgia Department of Ag, then Georgia Fruit and Vegetable Commodity Commission. I really appreciate all the support from the Commodity Commission and Georgia Department of Ag. Then Innovation Gateway, that has been very generous to give us infrastructure support, some of the funding support, especially when it comes to molecular breeding, because it takes a lot of money to do the molecular breeding. So that support has been very critical, so I appreciate all that support. So with that, if you have any questions, I'll be happy to answer. I try to keep it brief so I can answer some questions. If not, then I'll let other speaker to wrap up. But please do visit the poster outside.
Yeah, thank you. All right, so our next speaker is going to be Dr. Diefenbrock. Yep. And she is going to talk about the Snail management update. So,
I'm going to use the stand because I talk with my hands, and your microphone's going to go way the heck out of here. Okay. All right, so I am actually a citrus entomologist. So why the heck am I in a vegetable session? Well, we're one of the few groups that's actually working on this snail, and it's becoming problematic throughout much of the southeast region. And it looks like it does a lot more damage to vegetables than we currently see in citrus. So I'm here to share what we're learning, where we're at, and hopefully some of that can help you guys with your management needs up here, because it's a lot of work to screen chemistries, and snails are just a pain in the butt. If anybody's tried to kill them, you know they're just awful. I also have my contact information here, and I think on the last page, so if you have questions after this, you can always contact me, because we are going to scoot back to Florida after this talk, as lovely as Savannah is. All right, so just a little bit of background for those of you who have not experienced the snail yet. Bulimulus bonariensis is the scientific name. There's actually 2 scientific names that are floating around. We think it's a species complex. We're not sure, but we're going to go with this one for consistency. It has a couple common names. It gets called the ghost snail or the peanut snail, which we really need to get rid of. It really negatively impacts the peanut growers, but for some reason that name got adopted. So they're pretty big. They tend to be between 3/4 of an inch to 1 inch once they have a shell. And then they kind of grow bigger and bigger. And most of the older ones tend to be around 1 inch to 1.5 inches. And I don't think they get any bigger than that, but we really don't know. They're light brown to tan. They have what appears to be an aggregation behavior, but we're actually not sure if it is true aggregation or just the fact that they're all trying to get off a hot ground surface and they wind up in the same spot, and it looks like aggregation. And we also know, we're not really sure where the heck the population really came from. We know Argentina is suffering with it right now, so we think it's somewhere in the South American continent as the origin, but not 100% certain. And we think there's probably been lots of introductions over time. This is a lot of we think, because there's not many of us working on it, but a lot of people are impacted. So again, the general group of snails, Bulimulus, we actually have several native Bulimulus in Florida. So for us, it's really, really important how we manage the snail, because we don't want to impact our native snails. Most of the snails from the genera are from Central and South America and even the Caribbean. And they're referred to as tree-dwelling snails. For us in citrus, our biggest challenge, and that's actually a citrus tree, and that's a microjet right next to it, they like to climb up structures, and including our microjets. They go there for water because it gets very, very dry. We are actually in drought most of the year in Florida, despite the hurricanes and other water sources. We're in drought most of the year. So they'll clog our irrigation jets because they're looking for moisture, and they're trying to get up off that ground. And then one of the ways that they're getting around is we do see them climbing and adhering to equipment quite a bit. So they're very easy to accidentally move around. And I do include a note on here. On that first slide, I mentioned that there might be a species complex. May or may not matter, but sometimes when you have a species complex, they behave differently, and that can really impact your management plans. So this map is a little out of date. I think it's about 2 years old at this point, but I haven't gotten a newer one. And it can give you an idea of the distribution as of 2 years ago. But as of last year, we knew they were all the way up into Kentucky. So they've spread pretty far. And all the red dots are this particular species, bonariensis. The green is, I'm not even going to try to say that name. And the blue is guadalupensis. We don't see problems from the other two, but we do see quite a few problems from bonariensis. Sporadicus is the other name. I think the first time I ever published, I used sporadicus because that's what my State Department of Ag gave me, and the USDA mollusk specialist said, no, it's bonariensis. It's okay. All right, so how are they getting around? I already alluded to this. We don't know exactly how they got here originally. We suspect probably on shipping containers. The first time they were ever found was in Duval County, Florida, which is in Jacksonville. We've got a huge port there. A lot of international cargo going back and forth. So it would make a lot of sense that that could be how it got here. Again, not definitive, but highly likely. They climb up onto the equipment. They move field to field under that equipment. I've had growers move them underneath mowers, just not even thinking about it. They're like, all of a sudden, I've got these snails in my screened production house. Well, he moved his mower from the cow field to the cup structure, and now they're all over his trees in there. So we had to kill those. They can move on people. That's actually my leg. I was scouting one day and I looked down and there was a snail on my leg, so apparently they'll climb us too. And they're not just in ag. So there's another challenge here, is that they're in ag, urban, and natural habitats. And in Florida, we've had some problems with them going into our local zoos recently as well. And we don't really understand the whole context of what the snail is going to mean. For those of you that are familiar with giant African land snail, we have a lot of problems with that in Florida. And part of the problem with that is that it is a vector for a really nasty pathogen, a really nasty disease called rat lungworm, which is a nematode-based disease. This snail is currently being screened to see if it does or doesn't vector it, because we have no idea. So that could have a lot of human health implications, which is very concerning, especially in agriculture. So let's hope not. Let's just say, please don't vector it. We don't want that. And the next few slides are a compilation of things I've gotten from folks throughout the region. So to see some of the impacts on agriculture, we see product contamination, where they can actually get harvested in with your product, and that's going to be a problem. If anything goes to storage, snail comes out, starts eating your food. That's not good. They can impede irrigation, which is a lot of what we see in citrus. I've heard about damage to fleshy vegetables. And if you look on that bottom right picture, you'll see where it's actually causing problems with harvest. And we hear reports of them actually clogging up some of the harvest equipment, and that is hugely problematic. Then also in the residential end of things, this is from South Florida. One of our extension agents called me and said, I need some help. They're all over the side of this person's house. I don't really know what to do. And at that point, I was like, dude, I don't know either, but I'd probably just get a broom with a good stiff brush and knock them off and maybe squish them. I don't know. That was my answer about 4 years ago, because I really didn't know what the heck I was doing. I still kind of sort of don't. I'm learning. But we do know more about them now than we did when we first found them in 2020. In citrus, we found them in 2020. They've been other places before. So if you look, I hope this works. Do I get a little pointer? These are the eggs. The eggs are buried beneath the soil surface, and they hatch out. And there's no hatchlings here, or neonates as they're actually called. But they kind of just look like a little bitty slug. So you have a little bitty slug that comes out of there, and then as it consumes food resources, it starts secreting that shell. And then we'll see really tiny ones with a shell. I mean, they're maybe 1/4 of an inch tops. And they just keep growing and growing and growing. And they do need to consume calcium to get a hardened shell, which most of us put out calcium in our fertilizer mix. So that's something we might have to consider in our management program is, are there better times to apply that calcium that maybe don't help your snails but help your tree? That is many steps down the road. One of the things we have learned from having them in the lab for a while now, and I have snails still that have the original nail polish from a couple years ago. We don't actually know how long they live, because snails are great at persisting. They can get to this bigger size or a smaller size, and they can just close themselves up in that shell, and they'll just hang out till the conditions are good again. So that's one of the challenges with managing them. So we have no clue how long they'll last in the field. There was a laboratory study done in Argentina where they had about a 2-year lifespan in a laboratory with no predators, no pesticides, anything coming at them. So that's helpful, but we don't know how realistic that really is if you're in an optimal habitat versus hiding out in your shell, coming out, being exposed, hiding in your shell. No clue. The adults are hermaphrodites, so they can basically, they can be male or female as needed to reproduce. And they do require sexual reproduction. So they can actually get to really high levels of a population with just a few snails to give you a start. Some basic biology with their feeding. I've never worked on snails before these guys showed up, so we had a lot to learn. Snails are generally considered grazers. They usually feed on like the films on the surface of items. So like a decomposing plant's going to have all sorts of fungi and bacteria on its plant surface, and that's normally what most land snails want to feed on. Not all, but most. And that little cartoon shows you its radula, which is kind of like this little saw blade that goes over and over and over and over. And that's how they feed. They actually use that little saw blade, and then they start ingesting the materials from that. So if you see these weird rasping damage, that's these snails feeding or your other snails feeding. In general, most snails are going to prefer damaged or decaying vegetation, but we have seen these actually damaging fresh growing vegetation. The green stems of brand new citrus trees, the vines of grapes. We've heard about quite a bit on fresh fruit or fresh vegetables in this region. We also know that they're highly attracted to the moisture in those agricultural settings. We've been doing trapping in several fields, and we don't really find so many of these snails in the field margins where it's drier, despite the fact that there's plenty of things for them to eat. They tend to be in the field where we're putting out irrigation every morning. So there is a draw to the irrigation. And yeah, we heard all these reports of different damage. And so hopefully more people will be able to work on the snail going forward because there's only so much one lab group can do. Actually, two lab group. There's one up north that's doing a little work too. I brought in a friend who runs a biocontrol laboratory to help us understand this organism a little bit better and try to look for some predators because that would be really beneficial towards managing. So Nicole Quinn, who's pictured there, she is an assistant professor over at our biological control facility. And we're trying to just figure out what the heck these things are doing. So in a study, her student actually started looking at what was in the gut contents of these snails because we really weren't sure. These is just purely in citrus groves. We weren't sure what they were doing out there. Are they really using citrus or are they using something else? Is citrus just kind of being impacted because that's where they live? And if you look just briefly on that list, they're all weeds. The only thing on there that's citrus is the last couple items on there, the pomelo, grapefruit, tangerine, and mandarin. So they consume some citrus in our groves, but for the most part in our fields, they're going after weeds. So that helps us a little bit. Doesn't help our irrigation situation, but it helps us think about what they might be using in the habitat. And I strongly suggest if anybody in a vegetable system can get a hold of funding to do something like this, to reach out to Nicole's group and try to figure out what they're using in your system, because that could be used long-term to actually develop better management as we learn more about them and learn more about our tools. And eventually we do plan to actually use this information to develop our management plans. So I mentioned snails can survive a lot. All right, so if you're looking at a snail, they have this little fleshy cover. It's called an epiphragm. And so if you flip it over and it's dried up, there's not out there and active, a lot of times you'll see this kind of shiny surface on the inside. It's like closing a door. They basically can hide out inside of that shell through all sorts of nasty conditions, the heat, the cold, anything you want to spray at it. And so they have a great way to hide out. And if you look at the picture on the far right, that's what they look like when they start coming out of their shell. This is when they're actually susceptible to things. So when you find chemistries that work, you got to get them when they're active. This was a lesson that a grower learned, a kind of expensive lesson. So we learned with him. And we also know that a lot of snails and slugs actually are able to detoxify many, many, many chemistries. So that's part of why we're testing everything we have available to us that's currently labeled for use in citrus. And some of the chemistries I'm going to show you guys will and won't transfer. So I have all the active ingredients for you. You can look at any of that stuff. All right, so developing management plans. The three things that we're looking at is figuring out when they're active in your region and crop, the timing of daily activity, because we now know that they've got to be out and active and chemistries need to contact that flesh, and then what tools do we have? So right now my lab is mostly working on the pesticide end of it because it's kind of the fastest way to get the problem under control while we get the long-term solutions. One group has done some work with tillage and exclusion, and then my colleague is working on some biological controls, which is going to be much further down the pipeline. All right, so most snails are going to be active in the evening and morning periods, especially in the southeast because we get hot and dry during the day. So this is really important when you're thinking about management timing. So if you have a foliar chemistry that works, if you spray it once it's hot and dry, they are hidden up in their shell and you're not making any contact with the organism. The chemistry that works the best for us, which I'll show you in a few minutes, is abamectin. And my growers found out very quickly that if you spray in the morning when they're active, they die. They literally fall off the plants. But if you spray late morning when they're closed up in their shell, nothing. You just wasted your chemistry.
Did you say abamectin?
Yeah, abamectin. Right? It's a miticide. Don't ask me how. I don't. We haven't seen anything out of copper yet. I want it to work. But we already spray a lot of copper in citrus, and so we have a lot of copper in our soils. We have to be very careful about that. So that is something that we always have to keep in the back mind. Other systems, you might be able to be more liberal with your copper. We are actually planning on looking at more of the fungicides too this spring, especially that have some copper in them, so stay tuned. I know, but I need it for my mites too. It's cheap, I like that. So thinking about how you sample, there's some different ways to sample via passive trapping or active sampling to figure out your seasonality of these organisms. And these can be used as monitoring tools because you don't want to treat if you don't need to treat. Oh, I like that little 10-minute sign. That's handy. So active sampling, Silvana Paula Morez, who's up in North Florida, her group has done quite a bit of work with this active sampling. They have annual cropping systems in row crops that they plow under every year, so they can do different things that I can't do. I work in trees, the roots are right by the surface, so I can't do certain things like tilling. But they were using beet cloths, plant inspection, soil inspection in this first study, and they found that using a beet cloth, and I believe this was under peanut, they would get a greater capture of snails. They get a better idea of what's going on in their field to see snail activity. Now, this doesn't account for anything in the soil. We don't know what the heck's going on below ground yet. We're just now, my colleague figured out a rearing protocol, so we can just now start working on the juvenile stages of them. And it's surprisingly slow going to figure these things out. She also looked at roofing shingles versus commercial baits versus pitfall with beer, which is kind of the home trapping for slugs, and a cheap cardboard trap with water under a rock. And lo and behold, that cardboard with a rock on it actually worked pretty darn good because they're attracted to that moisture. So if you have a moistened piece of cardboard with a rock on top of it, they will hide out in that area, especially as the soil starts to dry up. They'll go there, and you can start trapping snails and getting an idea of what your population looks like. Is it perfect? No, but it gives you an idea that they're out and active. In tree crops, like I said, we have a little bit of a different scenario, and we notice that they climb a whole heck of a lot more. So we were trying to take advantage of the climbing behavior, and if you look in the top picture, you'll see this pyramid trap. It's literally just like two triangles put together and in the ground. We use something like that to capture Diaprepes weevils when they fly around. We have a trap that goes on top of it, and they've been clogging up all my Diaprepes traps. So I was like, cool, we'll try this. And then we had a flat version of the same material on the ground to see which one might work better based on some stuff Silvana had done and some of our observations. And one of the things that we found out is that the trap type in our scenario does matter for detection, just like it did in hers. But we think in our scenario, it's more influenced by ground cover. Our citrus fields that have ground cover, like purposeful cover crops or just a bajillion weeds, the flat traps actually attracted the snails more so than the pyramid traps. I think it's because they didn't have other surfaces to go up. And then the fields without, with the ground cover, the fire ants are a major problem with those flat traps. They love, if you've ever been in the field and you have a trap down there, they love that. So if you use these, just tell your tech staff or your scouts, be careful of fire ants.
Do the fire ants eat the snails?
No. And if you smash them, they do. You got to smash the shell. It's the only way they're in my apartment complex, so on my evening walks kind of mess with the snails. And yeah, just
curious.
Yeah, I was curious too. I was hoping, but no, they just like, they're when they're closed up in their shell, they kind of come up and antinate on it, and they're like, do you do anything? Hello, who's there? And then they leave. But if they're smashed, they'll eat them. Or if they're sick and hanging out of their shell. So there are some chemistries that will make them kind of start foaming and hang out of their shell, and then they're vulnerable. And in that period, yes, and my experience with most of those chemistries is that within 3 days, the snail was fine. So my first assays didn't go so good. They've been a real challenge. And then in the field without or with minimal ground cover, we actually had those pyramid traps worked really well, kind of because it was a surface just like the trees for them to go up. But we don't know like which one's going to be the best option for this spring. However, that said, our patterns, and I apologize that yellow line's a little hard to see. Hopefully you can see it okay. This trapping started in the spring of 2023. And we start seeing the snail population going to the traps and going up in this like June period. If you had actually been in the field, they were actually super active right in the period right before that. And we just didn't have the traps out there yet. And it kind of decreases as it gets really, really hot. We usually get some nice rains around here. They start dispersing around the area. And then it gets really dry right here. And we see more on the traps. Shock of shocks, they're going to the wet spots. And then it gets dry again, and then we go into 2024. It's cold. We're pretty sure they're underground because we don't really see them, because as soon as it warms up, you see them. But when it's cold, they're nowhere to be found. So in this 2024 period, they're kind of here, but then they really start getting busy for us in this March period. We'll see them all over the place. Thank you. And they just kind of keep increasing. So we have this cyclical thing going on. And we think that the trap capture pattern is pretty consistent with the wet-dry period. If it's wet and moist out, they don't need to be on those traps and get up off the soil as much. Additionally, we've seen in some of the cover crop fields where they're just active all day long because it retains so much moisture in the row center. Usually there's less of them on the irrigation, which is kind of cool. But we haven't like formally evaluated if that's going to make a difference. All right, so we're going to move into the timing of them. I already mentioned their activity tends to be tied to ambient temperature and moisture levels. So they like to get active kind of in the evening as the temperatures are, in the summer anyways, cooling off a little bit. It's getting a little more humid in the air. And then when it gets darker and the sun's not beating on them, they appear to be active overnight based on some red light camera trapping. And then they are active in the morning until it gets hot. And then when the soil starts heating up, they just beeline for the trees and get up off that soil. So that's helpful for us in thinking about how we manage it. Doesn't translate as well to vegetables, but they will likely go up the stalks of the vegetables. And feel free to correct me if I'm wrong on that. All right, so here's the most important part that you're probably interested in is really the chemical management updates. And I showed some of this last year, and we changed how we did our assays because the hurricanes kind of messed us up. So we found a better way to test all these things. Essentially, the most important thing is right here. One of my growers called me and said, hey, I oversprayed and my Agriflex hit them on the wall of my cup's house and they're just dead. So Agriflex is thimethoxam and abamectin. And I was like, well, that's really cool because nothing I have is working. So we went back and looked at Agriflex, thimethoxam, and abamectin in comparison to, of course, an untreated control. And it turns out it was the abamectin, which is that Agromec SC product, that had the highest kill rate of any of my foliars that we've tested so far. And these are most of the chemistries that we had available to us at the time, and then we will be doing some more fungicides and a few more foliar chemistries this spring. But the trick is, like I said, got to make contact with the fleshy part of the snail. If you spray in the afternoon, there's no activity of the residue. It's got to be hitting it right when they're active. And then the baits. Okay, we're really excited about the baits. Historically, we managed mollusks with baits. But most of the really efficacious baits are metaldehyde-based, which have a really broad spectrum of impacts. Metaldehyde affects any mucus-producing organism, so those are going to be your deadlines, the slugger. There was one more, but I don't think it's on this list. So like the really efficacious deadline here and deadline there. But more recently, a newer technology company has started working with us, and they've been developing some new baits for us. And you see they're labeled A1 right here, and it kills better than anything else I currently have. A2, pretty darn good. I can't tell you all the active ingredient yet, but they're getting close to field trials. So if anybody has a heavy population and an extension agent or researcher willing to work with you, I'll put you in touch. But it looks really efficacious, and I'm excited to see what comes with that, because a lot of the other baits have some issues with environmental concerns. Okay, do I have time to go into tillage, or should I pop past this? Oh, okay, tillage can help. We're working on some biocontrols. There are a couple things that seem to eat them, but they have to be out and active or damaged. I'm going to skip the mites. It's cool, but we'll see if this actually matters next year. So yeah, your basic things, we have some chemistries available. We've got some information about daily activity patterns, and you need to know a little bit about their seasonality when you're planning. And this is just a side note at the very bottom is, we know that it's in this area, and we know that it's marching up through the southeast. So if you're working with researchers and they ask for your support or they ask for a place to work in, please, please, please work with them. These are an absolute pain in the butt. And we need active populations. We need folks who can support the researchers with letters of support, space to work, whatnot. I'm probably like soapboxing this, but I've been very, very fortunate that I've had that in my system, and it's really made the difference in getting some quicker solutions out. Lots to acknowledge: commodity group, technical staff, other faculty who've worked with me, and I'm probably out of time. But my email address is really easy. It's just L, then that last name in the program book at ufl.edu. Just email me. I'll share any of this with you. Am I done? Okay. And we're going to roll, so I'm sorry.
All right. Well, in that case, we're going to roll on to our next speaker. We got Dr. Fonza. He's going to give us an economics update. So let's roll.
Good afternoon, everybody. I know you all have heard enough about pest and disease and stuff, and I'm going to be saying some crazy things here you don't like to hear. My name is Greg Fonsa. I'm going to be talking about the global and U.S. fresh food industry outlook. Here are some of the topics I'm going to cover. I'm going to talk about world vegetable situation and outlook. I'm going to talk about U.S. agriculture trade and trade balances. I'm going to talk about the U.S. vegetable and pulse outlook. And I'm going to talk a little bit about Georgia vegetable industry trend. And then all of us are going to draw a conclusion. In 2025, import of vegetable is projected as $53 billion. Gross vegetable production value is going to be $0.75 trillion, no, $0.7 trillion, with a 2.5% projected growth rate. On an average, global per capita vegetable consumption is 186 grams, which is equivalent to 0.41 pounds per day. WHO recommends 400 grams per day, which is equivalent to 0.9 pounds or 0.4 kilograms. If you look at the average fruit and vegetable consumption per income and region, you find out that between sub-Saharan Africa, Asia, Latin America, and the Caribbean, we look at three different income brackets: the low income, the average income, and the highest income. The red line in between here is the recommendation of the WHO, which is 400 grams per acre, per day, which is equivalent to 0.9 or 0.88 pounds per day. You find out that in the low income brackets section, which is on your, probably on your your right hand side. Yeah, your right hand side. Almost all the countries are below the average recommended intake per day. The yellow line is the yellow color is the sub-Saharan Africa, the red one is Asia, and the green is Latin America and the Caribbean. I assume that the U.S. is also included in this, this green. And then if you look at the average in the middle, you find out that only the Latin American country and the Caribbean are close or meeting the recommended daily intake. But then if you look at the right-hand side for the rich folks, you find out that the Asian countries and the Latin American and the Caribbeans are the ones that are above their daily intake, whereas sub-Saharan Africa is below. So now let's look at the production value and forecast. You see that from 2008 to 2029, in 2018, we were still having 0.9 trillion, 0.56 trillion, sorry. And that has risen consistently all the way to 2029, which is a forecast. It's going to be $0.78 trillion. That is because we are calculating 2.5% growth rate from 2025 to 2029. So you can see how that trend is going all the way up consistently. And now let's look at the agricultural production challenges. Number one challenge is climate change, which affects crop yield, soil fertility, and water availability. Also drought, floods, extreme temperatures, and fluctuating rainfall patterns. We have even witnessed this recently where we had the hurricane, so that also falls under these temperature issues. Technology adoption, e.g. precision farming, drones, artificial intelligence, which can increase yields and reduce costs and improve efficiencies. Let's look at the global vegetable trade and projection. We keep talking about global because what? We like to talk about globalization, and these days it's difficult to talk about one country per se, without leaving the rest of the world out. It's just difficult. So if you look at the import-export, the good part here is that the export is the red line is higher than the import, but both import and export of vegetables are all consistently climbing up, rising. So which is a good thing, means people are thinking about living healthy and eating good. Now, let's look at the market and the trade challenges. One, there's limited trade and market access. We'll be talking about tariffs, quotas, and other trade barriers that can limit access to global market. It can also reduce profitability and agricultural production. Increased competition from low-cost imports can undermine local production and threaten food security. And this is one of the reasons why the policymakers always talk about blocking the barrier between Mexico and the U.S. That's one of the reasons, because it can threaten food security. Now, wars and conflicts around the world is also a big issue. It's difficult to move things around when there are wars. Right now, if you want to fly from Europe to Africa, the plane has to deviate, go around because of Mali and all that kind of stuff. It costs more. Everything is a challenge. It's a huge challenge for everybody. Let's look at the U.S. agricultural trade and trade balances. This is not really good news, because if you look at this table here, even before this table, for the past 20 years, we have, when I say we, I'm referring to the U.S., we have always had a positive trade balance until 2019, where we finally got $1.3 million negative. $1.3 billion, that's a lot of money. And then, of course, 2020, everybody understands we had COVID, that was given. It drops to $3.7 billion. Then we did very well in 2021, and then 2022, and up to now, we are getting a big, big negative balance of trade. 2023 was already $17 billion, and as we speak, it's already $31, $32 billion. And by the end of this year, it might be worse than that. So that is not good news at all. Why is the U.S. ag trade failing or falling? One, the strong dollars. I don't know, those of you who send money abroad, you can see that our dollar now is very strong. The last time I sent money to a French country, I had it used to be 500, $1 US to 500 French CFA francs. Now it's about $1 to 630 CFA francs. So you can see how strong the dollar is. And when the dollar is that strong, it hurts us also, because those folks overseas cannot afford to buy our goods. That's the problem. It's both sides. You know, it has advantages and disadvantages. Strong competition from other agricultural producers like Brazil. They're giving us a good fight. Declining commodity prices globally. Increased transportation costs. I just mentioned a case where if you want to go to Africa now, maybe to Cameroon, the plane has to go around because there's war in Mali. They are afraid of this airspace, and what happens to transportation costs? It goes up. So if you had your strawberry in that plane, what happened? You're going to pay double because of the same issues. Lack of new trade agreements. We talked about that, and that has been in the past administration. We did not renew some of our trade agreements. Consumer preference for year-round produce selections. You know, if you go to the supermarket now, people want to eat mango. People want to eat tropical fruit. So bananas. So that's another thing, you know. Americans who have traveled abroad, they have acquired foreign tastes. So when they come back home, they want to replicate that. Oh, I used to see this in Indonesia. Oh, when I was in China, I saw that. Or when I was in Tanzania, I ate this. Oh, we have here in the market. So all those things come into play. U.S. agricultural trade and partners. The leading U.S. agricultural export are grains and feeds, soybeans, livestock, tree nuts, fruit, vegetables, and other horticultural produce. However, the leading U.S. imports are horticultural and tropical products. So you see the difference. Canada, Mexico, and the European Union and the East Asia are our major trade partners. So when there's any disruption in any of those areas, it affects us directly. Now, we talked a little bit about the trade agreement. You remember we changed NAFTA to USMCA in 2018. We refused to sign the Trans-Pacific Partnership Agreement in 2016. We also refused to sign the EU Comprehensive Transatlantic Agreement in 2013. So That's one of the reasons that it's affecting us. It's affecting the whole world as well. For example, Senator Kevin Cramer of North Dakota, this is what he says, I'm going to quote. The current sharp decline in U.S. agricultural export is directly attributable to and exacerbated by the unambitious U.S. trade strategy that is failing to meaningfully expand market access or reduce tariffs and non-tariff barriers to trade. That's a strong statement, and it's just the truth. Each time you put a barrier or trade tariffs simply means tax. The other country is going to fight back. No matter what you do, they're going to fight back. And when they fight back, what happens? They say when the elephants are fighting, this is an African adage, the grass is the one that is dying. So that's the problem with tariffs. Now, let's switch to vegetable. We are here about vegetable. Let's look at vegetable trade and the trade balances. As a matter of fact, vegetable trade and trade balances has not been as good as the overall agricultural trade. We have never, when I say we again, I'm referring to U.S., we've never had a positive trade balance. Now, I decided to go as far back as 2016. You can see that it was $6.1 billion short, negative. Now, how do I get this green line at the bottom? This green line at the bottom is the red line, which is export minus import. So when we calculate what we send out of, we sell to other countries, minus what we bring in, is what gives you the green line at the bottom. So usually when you buy more things than you sell, you are negative. The good part is to sell more than you buy. So that negative line means that we are buying more things from other countries and bringing it to the U.S. than we are actually selling to them. So you can see how that line is going, and it's widening every day, every year. In 2024, we have $12 billion shortage in the vegetable industry. It's totally different from the general agriculture, because the general agriculture includes things like heavy equipment and all those kind of things which some of the countries cannot produce, so they have to buy it from us. But now even the general agriculture is getting hit. It's getting negative. So vegetables is really in trouble. We have to step up the game. Let's look at the harvested area, for example. The top line here is the bottom line that I want to see. You can see that it's been trending down since 2020. Of course, we can always complain about COVID. It's been trending down to 2023, where it finally went up. And in 2024, it finally went up. You can see the stroke from, if you look at the green line in the middle, the green line actually represents dry beans, which we don't grow much in the southeast area, including Georgia. But you see there was a big jump in 2023 to 2024, which helped to increase the overall harvested area. But if you see the blue line is fresh and processed vegetable. You can see that it's just consistent. Straight line. It doesn't go up, doesn't go down. So what are we going to do? We have to make sure that that starts going up as well. Then, of course, the red line is potatoes. Just a few minutes ago, or an hour ago, somebody was talking about growing potatoes in Georgia. I think it was Dr. McAvoy who mentioned that. So you can see also potatoes is constant. It has not changed for all these years. Let's look at also the production. You see the three lines at the bottom? The blue line is fresh vegetable. You see how consistent it is? Fresh vegetable from 2020 all the way to 2024. You see how the red line, which is also processed vegetable. Before, we used to be really good at processing because we can sell that overseas, but because of the strong currency, people cannot afford it. So you can see that also sloping down. Then the green line is potatoes, which we just mentioned. But the overall is on top. Is that purple? You can see that there's a drop between 2023 and 2024. You can see the slight drop also there. So if you put all those three together, it gives you the one on top. Now let's look at the crop value. On your left-hand side, which is your right-hand side, you see fresh vegetables. Fresh vegetables for 2020 and 2021 was flat, constant, no increase, no decrease. Then it kind of bumped up in 2022 and 2023, and then it started falling again. And we're talking about the crop value. It started falling. Processed vegetable has been increasing until 2023 to 2024, it's slight drop. The only thing that stayed a little bit was mushroom, which we don't do a lot in this area here, but potatoes also started falling. But overall, when you put all the things together, you find out that we can see a slight at the extreme end here. You can see how the slope is going up. There's an increase. Oh, all right, I better speed up. And then here, the vegetable import versus export per capita consumption. You see that in 2020, there was a big spike. You see the blue line, big spike. Of course, everybody knows what happened, COVID. After that, it dropped. And now it's trying to climb again. It made a remarkable climb in 2023, and it's falling again. Let's look at the monthly price index. The monthly price index, the good part about this one is that if you see the dotted line at the bottom is the average of 2019 to 2021. But after that, all the prices were on top. The green line here, it's 2024. It was pretty high from the first quarter, then it dropped a little bit, then it went up in June, then it started dropping down. Now, if we look at individual crops, look at sweet corn, it's really hard to predict. The red line is 2024. It started off pretty good. That is the price percent. Then it just had a nosedive, dropped all the way down, and then started picking up again in July. Let's look at snap beans, similar situation. It's really hard to predict. So if you are talking to your farmers and trying to give them an idea of where to do, which plant to crop, which crop to plant, it's so difficult to tell them about the price because the price is so volatile. Onion was pretty good in 2024, but if you look at 2023, onion was in trouble, especially in the first quarter. Then it bumped up in June, and then it started dropping again. But in 2024, it started up on top, so you can say, hey, what happened here? It started dropping all the way down. Let's look at cucumber. Cucumber, similar problems, zigzag, zigzag. The red line is 2024. The green line is 2022. You can see how the prices go up and down, up and down. So the bottom line here is to be able to catch the right market window, which is not easy all the time. Look at tomatoes. 2024 was pretty good, but by June it had gone down, and then it started climbing again. So you see the prices are very volatile, and it's difficult for the farmers. That's why they are taking all the risks, but they are really good at taking risks because it's difficult for them to really predict where they're going to go. Let's look at the net export trend and processed tomatoes. The green line are the export, and the red line are the import, and the black line is the net import. In this case here, the export is better. And let's look at the farm, U.S. farm real estate per acre. Let's look at the bottom here. You have the United States at the bottom. You have the farm real estate value, average value, it's going up. The all crops is going up. And then the irrigated farms or plot is also going up for states, for different states like Alabama, Florida, Georgia is going up. But if you look at the annual cash rent per acre, you find out that overall in the U.S., for all crops, it's going up as well at the bottom there. For irrigated plants, it's going up, and for non-irrigated, it's also going up. But if you look at individual states, you find out that in Alabama, for example, it went up. For Georgia, it went up, but for Florida, it went down, and for South Carolina, it went down. This is the, let's talk about Georgia real quick. I hope I have one more minute. Georgia farm gate value trend. Despite all the problems that we have in agriculture, not only in Georgia, but in the whole United States, you can see that this industry is very resilient. Whether it's a hurricane, whatever it is, they still bounce back. When I came here, when I took this job, the farm gate value was only $600 million. By 2005, it had gone to $800 million. And by 2017, it gone to $1.5 billion, despite all the problems. And by 2022, it's $1.3 billion. So this industry, despite all the challenges, is very resilient. Now, let me show you a little thing here real quick. They're looking at me to shut up. If you see, for example, this table here, you have 2025 on your left-hand side or my right-hand side. You have 2012, you have 2017, and 2022. Let's take sweet corn. In sweet corn, it was 12% of Georgia farm gate value, 12.5%. Then by the time we hit 2022, it had gone to 14.4%. Now let's take another crop. Let's take a bell pepper. It was 9.6% in 2005, and then it dropped to 2017. It went up to 10%, and then to 2022, it's now 11.7%. Conclusion, due to the growing population, the increasing demand for fresh produce, and changing diet, the future outlook of ag-food marketing looks promising. Technological advancements are expected to increase productivity and efficiency of the sector. Agricultural sustainability will become a major concern. And I better just call it off, say thank you here, and I'll take any question you may have.
Okay, real quick. All right, well, thank you, Dr. Fonza.
Thank you,
Here's
sir.
your
Thank
mug
you.
as speaker's gift.
Thank you.
Appreciate you coming.
Yes, sir. Thank you. Can I take of this?
Yep, our next speaker is going to be Dr. Chowdhury, giving us emerging plant parasitic nematode threats.
Yes. Well, the good news is I am the last one, I think, right? So after this, you get to go home this Saturday. So that's the good news. Another good news is I'll try to make it quick. I know we're all about to fall asleep, but just a few more minutes, okay? So yes, I will talk about emerging plant parasitic nematode-related threats in Georgia today. That's going to be a majority of my talk. So just to show you the importance of plant parasitic nematodes. They are thieves. They're stealing grower yield, essentially. And it's a good amount of it. In the United States alone, we are losing $6 to $8 billion of yield in agricultural crops each year. And the extent of damage caused by, so it's not all caused by the same nematode. It's caused by a variety of different nematode, and the nematodes, the extent of damage they cause really depend on the type you have present in your field. For example, if you have root knot nematode in our vegetable fields, up to 50% losses are not uncommon. Whereas if you have a spiral nematode, damage potential is almost negligible at low numbers. Other things to keep in mind is host crop, nematode species, soil type, and environment. These all play a role in the damage that nematodes cause, and they play a role when it comes to managing them. So the most important thing still, I feel like, for nematodes is that nematode often escape management. And the reason behind that is because the symptoms caused by nematode are not unique. They mimic symptoms caused by any of the other things that you have there that reduces plant health. It could be foliar diseases, it could be nutrient deficiencies. So that is one of the ways they are successful at being the thieves they are. So it's very important to sample for nematodes. To that end, I'll talk to you about a survey that was started by my predecessor and kind of finished up by my team. We surveyed around 302 vegetable fields or fields with a history of vegetable production across 39 counties in Georgia. And we found root knot nematode, the most predominant nematode, to be in 66% of the fields, and about 14% of the fields had a mixed population of nematode. But interestingly, we detected 3 new species of root knot nematode in Georgia through the survey effort. We found guava root knot nematode in 2 fields. We found peach root knot nematode in 6 fields, and Texas root knot nematode in 2 fields. Now, a lot of you may have heard about guava root knot nematode. Georgia is not unique for having them. It's actually pretty widespread, if not predominant, in the neighboring states of Florida and North Carolina. So why is the scientific community so concerned about this nematode? Well, these nematodes can reproduce on crops that have resistance to the more common southern root knot nematode. So after a lot of effort, through a lot of effort, we have developed resistance into variety of crops, including vegetables, cottons, peanuts, etc. And guava root knot nematode can reproduce on those resistant crops / varieties, except for peanut. Peanut is different. So guava root knot nematode also has a wider host range, and this includes most vegetable crops and numerous row crops. And it's highly virulent. I'll talk more about that in a little bit. So let's get back to the map. Guava root knot nematode was detected in Tattnall County and Lowndes County. Now, Tattnall County being essentially in the heart of Georgia's world-renowned Vidalia onion region, the question came, is it a threat to the Vidalia onion region, Vidalia production of Georgia? And we didn't know if it even infected onions. So we conducted a preliminary study, and then we found that this nematode does infect onion. Then we conducted 2 greenhouse trials where we assessed the reproduction on different onion varieties, and if there is any difference between the varieties when it comes to their susceptibility to this nematode. And as you can see, that yes, the answer is yes, there is some differences. There, Tania and Vidora are the Tania, Vidora, Sweet Magnolia, Cepello, Rio del Sol, and Monja Blanca are Vidalia or sweet onions varieties. And Tania and Vidora were significantly more susceptible to Cepello and Sweet Magnolia. On the right there, we have Red Duke, Red Halen, Red Maiden. As the name suggests, these are red onion varieties, and NUN 1011 is white onion variety. And they were somewhere in the middle. When we looked at gall index, we saw a similar pattern, right? Some varieties performed better than others. Then we also wanted to look at the percentage reduction of fresh bulb weight. So just because it's reproducing, does it mean that it's going to reduce the weight, reduce the yield directly? So to show that, the answer is yes again. The black bars show where there was nematode, and the gray bar shows where there was no nematode. And as you can see, black bars are smaller, and so that means is that the fresh bulb weight when there was nematode was smaller for most of the varieties we looked at. We actually saw pretty significant differences with the red onion varieties and the white onion varieties. Red Duke, we saw 31.1% reduction of bulb weight. So to answer that question, can guava root knot nematode become a threat to Vidalia onion region? And the answer is it can, but it is not yet. We actually collected an additional 80 samples across Tattnall County and neighboring counties from onion fields, and we only found southern root knot nematode. Very good news. But it's very important to stay a step ahead of this threat, and we are. Having the differences in susceptibility in variety indicates that there is resistance / tolerance to these nematodes. So we just have to look for them. So there is hope. And some varieties are better than others. So let's move on. Let's say I talked about the southern root knot nematode. That's the most common one that we see here in the southeast. How does guava root knot nematode reproduction compare to southern root knot nematode? So to answer that question, we assessed about 9 of the major vegetable crops produced here in Georgia. And what we saw is that guava root knot nematode is the black bars. On the y-axis is their reproduction, and on the x-axis are the crops. And we saw for all of the crops, consistently, guava root knot nematode had higher reproduction. We also saw southern root knot nematode being much less than guava root knot nematode, and sometimes peach root knot nematode. Interestingly, pepper was not susceptible or was not a good host or moderate host to peach root knot nematode. So to answer the question, how does guava root knot nematode reproduction compare to southern root knot nematode? It has relatively higher reproduction. And what that means is that in a mixed population field, guava root knot nematode will outcompete southern root knot nematode and become prevalent. Pepper is a good rotational host for if you have peach root knot nematode. So the next question, is chemical management strategy still effective against guava root knot nematode? So ideally, we'd like to do this in the field, right? Testing the chemicals, but we don't want to spread it any more than it is. So we looked at them in laboratory and greenhouse, actually. And what we saw is that this table here shows the concentration you need to kill 50% of the nematodes. We tested vellum, cerebro, nimitz, vidate. For vellum, on the right here is the southern root knot nematode. You need almost 4 times the concentration to kill 50% of guava root knot nematode. 1.12 for southern root knot nematode and 5.30 for guava root knot nematode. This is in parts per million. Cerebro, though, was not as bad. For cerebro, we just needed a little less than twice the amount, a third less than that, more. Flu and cell phone, we needed a little less than, again, twice the amount. Oh, a little more than twice the amount. And then vidate, the numbers there were pretty similar. So cerebro may be a good choice for their management, but the other still does work. You just need higher concentrations. Fumigants, now these you really need to be in the field, otherwise somebody may have some health issues. So we looked at some of the work from North Carolina, where this is more prevalent, and what they found is that there's a slight decrease, but still telone, our major management tool, still good against guava root knot nematode. So the answer is nematicides are still effective. We may need higher concentration depending on the nematicide. There's also another good news. It's not all bad news, some good news too. As I said, we surveyed additional 80 fields, and we couldn't find guava root knot nematode again. We also surveyed 176 tobacco fields since we detected guava root knot nematode across 16 major counties of Georgia. A lot of these fields are rotated with vegetables, and we did not find guava root knot nematode yet. We processed a majority of these samples. We looked at 87 cotton fields across 10 counties, and again, no guava root knot nematode yet, but we haven't processed a good bit of them yet. So in conclusion, there are only few examples of successful nematode management in the world. I know of one in Idaho, potato cyst nematode, right? So this nematode has already become widespread in our neighboring states, what we can do is slow its spread. Prevention is still the best medicines. So nematodes only move short distances themselves. So humans are the ones that facilitate long distance movement. So it's very important to use clean certified seeds, slips, transplants, inspect them, get it from reputable people. Both the sweet potato fields, both the fields that we found guava root knot nematode were sweet potato fields that got their slips from outside the state. If you already have an infested field, rotate with non-host crop. I know the practicality is an issue, right? We can't always rotate with what scientists say because we need the tools to cultivate that. But regardless, there are some options. If you have peach root knot nematode, pepper and cotton, good choices. Again, if you have guava root knot nematode, mostly agronomic crops. Corn, peanut, sorghum, small grains are good choices for rotational crop because these don't reproduce on them as much. Nematicides, telone is still effective. Celebro and Vidate may also be a good choice for controlling this nematode. So let's move on. Let's move on to the existing threat. Just a few more slides left. So again, as I said, most abundant for us here and prevalent, widespread is southern root knot nematode. And when it comes to their management, telone 2 still continues to be the best management tool available to growers, the fumigant. However, I always hear in the beginning of each year, this year we might have some supply shortages. And I have been told that could be a tactic by the companies as well. So I can attest to the truth of it. But chemicals has been phased out previously. So with all these things in mind, as I said, vegetables, especially tomato and peppers, are one of those crops that have resistance to root knot nematode, southern root knot nematode. But most of our growers here don't know about them. And so it came to my mind, it's like, why is that? To answer that, we conducted a trial. It's a preliminary trial, essentially, but we conducted a trial with 5 replicates, and this is the results. Can you tell which were the resistant varieties? It's pretty simple. The 2 with the smallest bar are the resistant varieties. These are the nematode counts. We did not see a statistical significant difference. That's because our population was low in our research field. When you want these nematodes, you don't get them. When you don't want it, it's everywhere. So that's the life of a researcher. But we also looked at these varieties. So a little bit details on it. Seminis recently released a resistant pepper variety. Charleston Bell is an older resistant bell pepper variety. And Aristotle is a grower standard that's commonly used. And California Wonder is another susceptible bell pepper variety. We looked at them, these 4 varieties. So this center red bar is the data I just showed you in the previous slide. To the left of that is same varieties with fumigants, and to the right of that, same varieties with vellum. And you can see when you're having the resistant varieties, you may not need these chemicals as much. But there is a lot of other questions. Just because it does well against nematode doesn't mean it's perfect. So I'm collaborating with Ted McAvoy there, our extension vegetable production specialist. I don't know if anyone would be better than that to let us know what is the yield potential there. And we'll probably work with the economist as well to determine if it is something practical for our Georgia growers. So this is going to be a future focus for my program, essentially economics of these resistant varieties, also optimizing non-fumigant nematicides for management of southern root knot nematode. For the emerging threats, when I started, we didn't have our toolbox, management toolbox was empty. I feel like with the past 1 or 2 years, we have some things in that toolbox. So now we are moving to more towards southern root knot nematode. Lastly, just one slide, and then this is the last slide. I have heard some questions and comments about pine bark, mainly from the northwestern Georgia area, mainly from homeowners, home gardeners, not as much from the conventional vegetable growers. Does pine bark work in controlling nematodes? There are some reports of pine bark mulch helping nematode control, and that's coming more from the home gardening world. I've been looking at trying to find research papers, and it's not as prevalent and as robust of a comparison. Still, I tried to do a small study to look at that. Pine bark does have alpha-pinene and D-limonene. These are insecticidal, pesticidal compounds. D-limonene is what you find in your home pine sol that you use to clean your home. So I evaluated water-soluble pine bark extract, essentially soaked the pine bark and then used that extract to look at nematode mortality. And what we found is that it didn't do anything to the nematodes. We also looked at directly these alpha-pinene and D-limonene. It didn't work. So does pine bark help control nematode? Well, at least we can tell pine bark extracts do not suppress root knot nematode. But pine bark mulch, even if they don't directly suppress nematode population, what is likely happening is adding the pine bark mulch is changing your soil physiology and changing the microbial community. As a result, nematode population, plant parasitic nematode population may be getting suppressed. So we are testing that a little bit more controlled way, and maybe we'll talk about it more next year. So next year it will be, so stay tuned, come back next year, and hopefully I don't know if I'll be the last one, but I'm happy with it. But we'll talk about these things next year more. With that, thank you. Nabeen is my graduate student who worked with the emerging nematode, plant parasitic nematode issues. Yes. Any more questions? Any question? How did I do on time? Yes.
What about, mustard as a biofumigant?
A lot of work has been going on, and there has been studies that showed it does significantly suppress. Mustard, you caliente, right? You introgress that. And I've been also working with Babesh a little bit on that. But we have to understand, you're not competing with telone with mustard.
Right, exactly.
But it does work. It's an additional tool. If it fits in that your production program, it's going to help. But another quick thing to add, just because it worked, let's say, somewhere in the Midwest, with a variety from there, doesn't mean it would work here in a Georgia's farm. So trials, research from here, around Florida, South Carolina, that would help make decisions on what would work. Not all mustard would work. It depends on the type, variety of that mustard.
Thank you.
Any more questions? Thank you.
Well, I appreciate everyone hanging in there with us, and I guess probably a lot of y'all are heading home today, so have safe travels. And I know y'all love filling out surveys, so there's a QR code. Also, there's paper copy if you would like to do that, but definitely feedback always helps. So thanks, everyone.